mycoplasma, and also PPLO. What do you mean by PPLO? That is pleuropneumonia-like organisms. All these will come under the examples of prokaryotic cells. Coming to some general characteristics, they are the cells that have no membrane-bound nucleus. These cells have no membrane-bound nucleus in them. Okay, so these have very simple structure and they are very primitive cells. They are primitive cells and they are also simple. They have no membrane-bound organelles or nucleus. The nucleus in them have no membrane around them. Examples of such prokaryotic cells are the bacteria, blue-green algae, mycoplasma, and also PPLO. What do you mean by PPLO? That is pleuropneumonia-like organisms. All these will come under the examples of prokaryotic cells. Coming to some general characteristics of prokaryotic cells, they are almost smaller than the eukaryotic cells. They are majorly smaller in size than the eukaryotic cells, and prokaryotic cells multiply faster. Prokaryotic cells multiply faster compared to the eukaryotic cells. So the prokaryotic cells show a great variation in their shape and size also. Coming to the shapes of bacteria, I have already seen there is bacillus, which is rod shaped, and then we have coccus, okay, which is spherical in shape. Then we have a vibrio, which is a comma shaped bacteria. Then we have spirillum, that is a spiral shaped bacteria. Okay, so their shape is related to their movement and function. Their shape majorly depends on their function and movement. Shape depends on their function and movement. Coming to the basic organization in prokaryotes, even though these prokaryotes have many different shapes, functions and size, they have a very basic internal structure which is similar. So they have basic internal structure which is maximum same in all kind of prokaryotic cells, that we see a cell wall and a cell membrane. We see a cell wall and a cell membrane in a prokaryotic cell. So cell wall is a boundary like structure that surrounds the cell membrane. This is present in all prokaryotes. We see a cell wall and a cell membrane in all the prokaryotes except the mycoplasma. Mycoplasma is a prokaryotic cell, but it has no cell wall. Okay, the cell wall gives shape and protection to the prokaryotic cell. What is the function of cell wall in this? This gives protection and shape. This gives protection and shape to the cell wall. Next, after the cell wall and cell membrane, what is the next thing we find in a prokaryotic cell? That is, we see a cytoplasm. Cytoplasm is a semi-fluid like structure that fills the whole inner part of the cell. What do this contain? Cytoplasm contains enzymes and ribosomes. Cytoplasm contains enzymes and ribosomes. Cytoplasm is a semi-fluid like substance that is present inside the cell. We have seen that cytoplasm is the site of all metabolic reactions. All the metabolic reactions occur inside the cytoplasm. Here you can see that there is a cell wall in the prokaryotic cell. We have ribosomes in the cytoplasm. The inner fluid like structures that are present are the cytoplasm. So next thing we see inside is the nucleus. Coming to the nucleus, these prokaryotes, they do not have a true nucleus. They do not have a true nucleus. That is, their nucleus is not membrane bound. So their genetic material, that is the DNA that is present in the nucleus, is naked DNA. Naked DNA because it is not covered by anything. We see the DNA like this, its coil structure here, so it is the naked DNA. So it is not surrounded by a nuclear membrane. Nuclear membrane is present only in the eukaryotic cells as they have membrane bound nucleus. Next, coming to its genetic material, it has a genomic DNA. That prokaryotic cells have genomic DNA. This DNA is single and it has a circular chromosome in it. It has a circular chromosome. Coming to the plasmids, they even have plasmids. Plasmids are small circular DNA that are present outside the genomic DNA. So the genetic material of prokaryotic cells have a genomic DNA, that is a single DNA, and also along with that we have plasmids which are present outside the genomic DNA, and they are small circular DNA. So these are not essential for the survival, but they give the special characters. These plasmids are not for the survival of prokaryotic cells, not for the survival of prokaryotic cells, but they are for the special characters. They give the cell some special characters, like example, some antibiotic resistance in a bacteria, such special characters are given by plasmids. These plasmids are used in genetic engineering. Plasmids are used in genetic engineering and bacterial transformation. Coming to the cell organelles of a prokaryotic cell, in the prokaryotic cell we have no membrane bound cell organelles. No membrane bound cell organelles are seen. That is, we have no mitochondria, endoplasmic reticulum, the Golgi apparatus, and lysosomes. So we don't have all these. The only organelle that is present in prokaryotic cell, that is only ribosomes are seen. Only ribosomes are seen in a prokaryotic cell. We know that these ribosomes help in protein synthesis. We also have some inclusion bodies in a prokaryotic cell. That is, prokaryotic cell have some special structures called as inclusions. What do you mean by these inclusions? These inclusions helps in the storage of nutrients. These help in the storage of nutrients, and also they help in survival. And the storage of nutrients. After inclusions, we have mesosomes. Mesosomes are the unique features of the prokaryotic cells. Mesosomes are very unique. They are seen in only prokaryotic cells. So these mesosomes are the infoldings of cell membrane. That is, after the cell wall, in the cell membrane we have such infoldings. These infoldings of the cell membrane are said to be our mesosomes. So how do these mesosomes help? The function of mesosome is that they help in the respiration, DNA replication, also in the cell division. So this is all about the prokaryotic cells, that we see that the basic internal structure of all prokaryotic cells is same. In a prokaryotic cell, we see a non-membrane bound nucleus, cytoplasm, genomic DNA, plasmids, inclusions, and mesosomes. So prokaryotic cells are small, simple cells without true nucleus, and no membrane bound organelles are seen, but they efficiently carry out all the body activities. Here you can see the infolding of the cell membrane. This infolding of the cell membrane is called as our prokaryote. We also seen that mycoplasma is the only prokaryotic cell that have no cell wall. Plasmids are the genomic materials that are present outside the genomic DNA. They are helpful in some special character developments. So next we will move into the cell envelope and its modifications of the prokaryotic cell. Next we will see the cell envelope and its modifications in prokaryotic cells. In the previous topic we have studied about the prokaryotic cells. Now we will learn their outer coverings and their cell envelope, and about the ribosomes and the other inclusion bodies that we see inside a prokaryotic cell. Even though the prokaryotic cells are simple, their cell envelope is chemically very complex. So they have a very complex cell envelope. What is the cell envelope of a prokaryotic cell? That is, in most prokaryotic cells, like especially bacteria, we see a 3-layered protective covering. This 3-layered protective covering of a prokaryotic cell is called as its cell envelope. So what are the 3 layers of cell envelope? The first layer being glycocalyx, and the glycocalyx is the outermost layer. After the glycocalyx we have a cell wall. This cell wall is the middle layer. After the cell wall we will come to see a plasma membrane. Plasma membrane is the innermost layer. In this you can see that the outermost covering is the glycocalyx, and inside the glycocalyx we have a cell wall, and the cell membrane being the innermost cell membrane is the innermost layer. So though each layer has a separate function, together they act as a protective unit. Each of these layers has some different function, but all the three layers together they help in protecting the cell, protecting the prokaryotic cell. Based on this, the bacteria is classified into the gram positive bacteria and gram negative bacteria. Okay, bacteria is basically classified based on the cell envelope structure. Bacteria is classified based on the cell envelope structure, and also according to the response of gram staining. Okay, based on its response to gram staining and the cell envelope structure, the bacteria is divided. So there are two types of bacteria, that is the gram positive bacteria. We have the gram positive bacteria and the gram negative bacteria. We have two types of bacteria. Gram positive bacteria is something, this takes the gram stain. When we stain with the gram stain, it takes the stain, and gram negative bacteria, it do not take any stain. It do not take the gram stain when it is stained. So this difference between gram positive and gram negative bacteria is majorly seen due to the cell wall thickness and composition. So why do we see this difference among the bacteria? This is because the composition of cell wall, its thickness. Okay, based on the composition of cell wall and its thickness, we have two different bacteria like gram positive bacteria and the gram negative bacteria. Okay, so this point is very important, please remember. So now first let us see about the outermost layer, that is the glycocalyx. We will go in detail about the glycocalyx. That is glycocalyx. What is glycocalyx? It is the outermost layer.
Doubts stay uncleared. Hours of self-study, yet biology still feels confusing. Because sometimes you don't need more lectures, you need personal guidance. At Crack Mentors, students get live personalized online one-on-one biology classes with dedicated faculty support, personal doubt solving, and focused improvement strategy.